In this video, we will discuss about replication compartment of SARS-CoV2. What does this mean? This means that where the viral genetic material replication occurs in case of SARS-CoV2 virus. So there, as we are so far we are aware about NSP, that is non-structured protein. So there are several NSPs in the SARS-CoV2 genetic material. So the ones that are essential for replication are NSP 3, 4, 6. What it does is that it converts the endomembrane of the intracellular into the replication organelles. Let's convert endomembrane or divert endomembrane towards replication of replication organelle. So now this endomembrane, which are mainly the endoplasmic reticulum and the Golgi body or Golgi apparatus. So these NSPs convert this endomembrane to function like replication organelle, and so far the studies have found that when in this organelle there is a formation of double membrane vesicle DMV, double membrane vesicles, and there is a huge variability of this double membrane vesicles. Convoluted membranes were also formed, and these are only formed during the virus replication cycle, or you can say virus cycle. In normal condition, this double membrane vesicles does not form, and in SARS-CoV NSP 3, 5 and 8 are found to anchor with double membrane vesicles. However, it is not yet clear where exactly translation of the viral RNA occurs. But what people have identified so far is that within this DMV there is a formation of double-stranded RNA. There is a formation of double-stranded RNA, and in that, and after that, this double-stranded RNA is translated into the genetic codes or the structure of the viral proteins that are encoded in the RNA. And there is recently there is another revelation that has been found in different virus. I'm not able to remember the name, but it might be MID, or you can check that name in the reference paper. But what happens is that NSP 3 has been bound to double membrane vesicles pore. The pore which binds NSP3 and NSP3 to double membrane vesicles has been found to span out these double membrane vesicles. So this has been found to span double membrane vesicles, and this kind of structure has also been observed in SARS-CoV2. Similar structure. So it is now hypothesized that within DMV the translation process of the viral genome occurs. So far there is no evidence has been found that genetic genetic materials is come out into the cytosolic part of the cells. And this viral replication occurs in a way that it does not get captured by cytosolic immune or as a sensor. So, however, the exact things is still not clear. But one recent studies have find out that for SARS-CoV2, NSP2 to 16 and nucleocapsid. Nucleocapsid contain most of the viral component. So this gives an motivation for the future studies this gives a motivation for the future studies to check the endomembrane response in case of check the endomembrane response in the presence of NSP2 to NSP16 and nucleocapsid, and how this endomembrane transform to a replication organelle. This part can be checked, and when this replication organelle function has been established, then they can check in the presence of RTC, that is replication transcription complex, that is which is present inside the viral genome. After this replication organelle function has been established, then people can check the microarchitecture, or we can say how this replication transcription complex behaves in the replication organelles or endomembrane converted replication organelles, and then we can monitor the several basic functions, that is translation, transcription of viral genome, transcription of viral genome in vitro, that will give us a more detailed idea or correct information about the behavior of SARS-CoV2 and where exactly the replication occurs inside the ER membrane, inside the cell, not ER membrane. And apart from this, one more thing that people have identified, the things which are mainly involved in SARS-CoV2 replication, or intracellular parts which are mainly involved in SARS-CoV2 replication is ER membrane, that we have already discussed, and early secretory stage of vesicle into the outside the cell, stage of vesicle. And the last but not the least is the trafficking of the vesicles outside the cell. This is the early secretory stage, that is when the vesicles are being decided based on its signature, whether they are to remain in the cell or be present as a transmembrane or secreted outside the cell. And the last step is the trafficking of checking of vesicles. That is usually after the trans region of the Golgi. That is usually happens after the trans region of the Golgi. So these three are mainly involved in the replication of SARS-CoV2. That's all for this video. Thank you.